So, cambium is, as I promised, the other type of meristem. It is a cellular plant tissue from which phloem, xylem, or cork is going to grow by division. This will result in woody plants in secondary thickening. So this is a plan diagram, which we'll be discussing in much more detail in Unit 4, but it's not a Cambridge-approved plan diagram because, well, it's shaded. And of course, as we know, there's no shading in plan diagrams. Now, we have here our outer epidermis. We have our cortex. We have our primary phloem in these stations here. We have our primary xylem here. We have our pith here. Oh, by the way, it's also not an acceptable plan diagram because this, we have a word written into the picture. Unacceptable. Also, there are errors on the ends of these label lines. Unacceptable. Now, here we have this black line here is cambium. Now, we can further divide this into fascicular cambium and interfascicular cambium because these are all fascicles, but these are vascular bundles. So each vascular bundle consists of xylem and phloem and xylem, with cambium in between. Because as cambium divides, because it is a stem cell, we have cambium here, and it will divide in this direction into phloem. And where'd my black marker go? And in this direction as xylem. So it will divide in both directions and differentiate differently in either direction. So in this direction, we'll get larger cells, in this direction, we'll get these larger phloem cells if this is the interior of the plant. Let's say that this will be our xylem over here. Here's the exterior of the plant, here's the pith over here. Here's the endovascular bundle. And over this way, we will get our two different cell types in our phloem, where we'll get our smaller, darker companion cells and our larger open phloem seed tube elements all mixed in together as we grow and divide that way. So that's the division that we're going to get. They're going to change as they divide. So they're going to divide and differentiate. And they're going to be forced outward that way as they divide and grow. Now, the goal of DNA replication. The goal of DNA replication. The goal of DNA as a whole is to store information for the production of primary protein structures. RNA is in that process, but RNA is also sometimes the goal, for example, for RNA and tRNA. So mRNA is a product that is used for like an intermediate. In order for proteins to function, they need to have a specific shape that's determined by their 3D conformation, their tertiary structure. So this 3D conformation through the tertiary structure, it relies upon the levels of protein structure beneath it, the secondary and the primary. The primary structure is a specific sequence of amino acids that forms the polypeptide chain. And if you change that specific sequence, then you completely alter the tertiary structure, the 3D conformation of a 3D polypeptide, the 3D conformation of a single polypeptide chain. So if you change the protein sequence, you change the protein's 3D structure. If you change the protein's 3D structure, you change its active site's structure. If you change the active site structure, you affect the way that it can do its job. If you affect the way it can do its job, then you affect, then you can change the trait. So changing the DNA changes the RNA. Changing the RNA may change the protein. If you change the protein, it may not function. If the protein's function was vital to the cell cannot perform that task anymore, and the cell will die. So DNA replication has to be what's called high fidelity. High fidelity is error-free. And if you make a mistake in copying DNA, effectively that could be a lethal mistake. There is a high risk of failure, and failure is an option that could kill you. Failure is always an option. It's just not a good option. So if you're copying DNA trillions of times, and any single time that you fail, you could die, those are pretty high stakes. So let's talk about the ways in which we replicate DNA. There were three competing theories for how DNA was replicated. We had the dispersive model, we had the conservative model, and we had the semi-conservative model. Now, the semi-conservative model is the one that ended up being the one that had the most evidence to support it, and therefore that is the one that we know and believe. Well, the one that we have evidence to that is truth, and therefore the one that is considered truth. So we have evidence that the semi-conservative model is true, so we believe it to be true. The conservative model and the dispersive model lack evidence, so we do not believe they are true. So, how is it that we came to have this evidence? We came to have this evidence through the work of Meselson and Stahl. Now this is a big nasty slide, but effectively what this is this is an experiment where they took bacteria and they grew them in an environment rich in nitrogen-15. Nitrogen-15 is an isotope of nitrogen, which is an element on the periodic table. In case you hadn't noticed, go look at one. I have a couple here. I've used it as a prop before. All right. So nitrogen-14 is the more common isotope, but nitrogen-15 is heavier and does the same chemistry. So nitrogen-14 and nitrogen-15. If we use nitrogen-15 and we grow bacteria in it, then they will form their DNA out of nitrogen-15. It will be heavy DNA. In a centrifuge, when we spin down DNA, the heavy stuff goes to the bottom. We will form a band of heavy DNA. So in the generation 0 that was born entirely in nitrogen-15 environment, 100% of their DNA will be heavy DNA. Now, if we take that bacteria out of the nitrogen-15 nitrogen-15 environment, and we put them into a nitrogen-14 rich environment, they will start to utilize that nitrogen-14 to build their next set of DNA. And what we got was 100% of the DNA in the next generation was 100% of a different weight of DNA. So, at that point, we knew that if 100% of the DNA was a different weight, it was either that nitrogen-15 was being converted into something else, which is not possible, or they were making one of an entirely new set of strands. So of these three models, we have the semi-conservative model, which took nitrogen-15 in pink, and the nitrogen-14 would be blue, half nitrogen-15, half nitrogen-14 in all future offspring of the first generation. That would account for our 100% band of a new density of DNA. The conservative model would not. We would have two bands. One that was all pure nitrogen-14 DNA, and one that was all pure nitrogen-15 DNA. We would have gotten two bands, one here and one here. So the conservative model is out in the first 20 minutes of the experiment. With the dispersive model, however, we're still not quite there yet. We have the dispersive model, the DNA of the parent is cut up into pieces and divided evenly among the offspring, and the offspring fill in the gaps for themselves. So here we have two strands that are different, but have the same density. So the dispersive model and the semi-conservative model could both fit this. However, the next generation had two bands. Half of the generation had the same type of DNA weight as the previous generation, which was this half and half, as we came to understand it, and the other had pure nitrogen-14, 50%. That does not compute with the dispersive model. With the dispersive model, we would have seen progressively lighter and lighter bands, all of the same weight, all of the same density. With the semi-conservative model, we do see that. We do see that after another generation, we have the conserved grandparental strand, as I'll call it. That's not what anybody else calls it, though. Is conserved here, and we have an entirely new nitrogen-14 DNA strand here and here. So, we have half and half of a new DNA strand. In consecutive new generations, we keep seeing this hybrid DNA generation getting smaller and smaller and smaller, and our pure nitrogen-14 DNA generation getting larger and larger and larger. That makes sense according to our semi-conservative model. So this is the evidence. This was the evidence that backed up with the strongest voice the semi-conservative model. And that's why we believe the semi-conservative model is true. Because we have really strong evidence based on strong physical understanding of physical principles. Now, remember how I said back in Unit 1 that DNA is a really long molecule? Why, yes, it is a super long molecule. If we started at one end of a DNA strand and worked our way to the other end, we'd be at it a while. If you talk about a human baby, you have about 40 weeks to go from one cell to about 1 trillion cells. So DNA replication needs to go faster than starting at one end and going all the way to the other end. You can't exactly be running from here to the moon, because that is literally what 1 trillion, actually, wait a minute. There's plenty of videos on the internet that tell you just how long your DNA is. I'm pretty sure you could stretch from like here to the moon a couple of times with your DNA in your cells, but I'm not going to look up that fact. I'm just not going to do it. Go look up on like Nerdy Facts or whatever. I'm not a fan of those websites or whatever, but 10 nerdiest facts. I'm sure there's a video somewhere. Clickbait. All right. But yeah. So you can't be doing that. You can't start at one end and go all the way to the other and just keep doing that. It's not going to work. So we're going to need to start at multiple starting points along the entire length of the DNA strand. So we start here, 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 and there, all along the strands. We start at multiple origin sites, and we open up multiple replication forks. These replication forks will expand in that direction and that direction until they meet up and we have two fresh DNA strands, well, half fresh anyway, DNA strands ready to go. So these replication bubbles expand, and these are replication forks right here. Now, do you remember how I said back in Unit 1 that new DNA strands only form in the 5' to 3' direction? Wow, that is from a long time ago. And it's a really specific detail. Indeed, hypothetical person watching this video, indeed. This poses a problem, because remember how I said that complementary DNA strands run anti-parallel to one another. That too is a very specific detail. But that one, I hope you remember, slightly better than the other detail. So, how are we going to run DNA in both directions of a forming bubble at the same time, if they both have to run in the 5' to 3' direction, how are you going to run an opening fork both in this direction if you are moving 5', 3', 5', 5', how are you going to do this? Because this one works. This one does not work. This one works. This one does not work. Oh, I take that back, hold on. DNA is formed 5' to 3', so this one would work. Backwards. How are you going to do that? Well, I have an answer for that too. This is one of my favorite pictures that I found on the internet. Educational use. Alright, so a summary of DNA replication. We're going to start with helicase. Helicase is going to unwind and unzip the parental double helix. So they're going to unwind. Then we have single-stranded binding proteins. They're going to stabilize the unwound parental DNA. They're going to prevent it from annealing back together. When I say anneal, I mean the DNA hydrogen bonds back to the hydrogen bonds that were just broken. The hydrogen bonds will reform that were just broken by helicase. So single-stranded binding proteins will hold that DNA, these two separate DNA strands, apart from one another. Even though that cytosine really wants to get back together with that guanine and those sweet, sweet 3 hydrogen bonds that it misses so much. So that's what's holding them apart. The easy part is the leading strand will be synthesized continuously in the 5' to 3' direction by DNA polymerase. DNA polymerase, DNA polymer ACE, it makes a DNA polymer. It's an enzyme, ASE, that makes a DNA polymer. It's named after what it does. Aren't scientists lazy? Alright, so it makes a DNA polymer. So it synthesizes that in the 5' to 3' direction, just as I promised you. So it synthesizes in the 5' to 3' direction. And the other side, which is the 3' to 5', we're going to have it also synthesized in the 5' to 3' direction. But that side is going to be the lagging strand. And the lagging strand is named so because of No, your computer isn't broken or messing. We're actually going to pause it there, and we'll get back to you after these messages. No, we're not monetized yet.